My name is Jeremi. I come here from one of the Harvard hospitals. By background, I'm a physician turned scientist and work with neural engineering. And I will talk about closing the loops in brain stimulation, so how we can record brain activity and how we can stimulate based on those recordings. To give us some outline, I will start by providing a whistle-stop tour through electrical stimulation of the brain and the body throughout the ages. Then I will give us some common language to talk about what an open loop is, what a closed loop is. And then I will talk about 3 different loops across different conceptual and temporal scales that we hope to close with brain stimulation to take it to the next level. And then I will talk slightly about where we may be headed. So to start with antiquity, 1st century AD, we had this Roman physician, Scribonius Largus, who used torpedo rays, which are a species of fish that can generate electricity. And he put them to the head of their patients to treat different conditions. So that would be sort of equivalent to today's electroconvulsive therapy. Then, 1700 years later, we had seminal experiments by Galvani, where he connected frog corpse to a wire that was looking out for lightning strikes. And those lightning strikes were generating electricity and stimulating the frog muscles, and the frog muscles twitched in response to electrical stimulation, which gave us an idea that perhaps there is something in our body that's excitable by electricity, and maybe we can do something about it. Then, in the 19th century, we had these 2 German guys who came in and expanded that concept from a humble frog to canine. And so here we see dog brain, and marked we have the areas where electrical stimulation evoked twitches in the leg muscles. Shortly thereafter, Penfield and Heath took that concept to human brain. Penfield was a neurosurgeon, the first functional neurosurgeon, as we call them today, in Canada, and he stimulated human brains across lots of patients. And he found some areas that, when stimulated, evoked motor responses, and some areas that, when stimulated, evoked sensory responses. Fast forward to the 1990s, we figured out that we can put wires into the brain. We can put those electrodes into deep structures, subcortical structures in the brain, in the basal ganglia, and we can stimulate the brain to treat different conditions. And it all started with Parkinson's disease, but now has expanded to many more applications across neurology and psychiatry. But this approach first was driven by where we want to put the wire. So we can see a stereotactic atlas. So this is an atlas of the human brain, where we can map it to a frame that's screwed to the patient's head, and we can insert those wires with high precision. But that was where it really ended. So we put the wire in, we turn the stimulator on, and we hope for the best. And that was until 2013, where we had the first therapy that was approved by the FDA that closed that loop. By closing that loop, we have the system now that records brain activity from one area, recognizes patterns that are abnormal, and then stimulates in another area to silence down those seizures or seizure-like activity. And fast forward to this year, earlier this year, we have an adaptive therapy. So what was developed in the 1990s, 35 years later, we have the adaptive or closed-loop version of it. So Medtronic was approved to use this system for Parkinson's disease. And now underway, there is lots of studies to expand it to other conditions. That was work done mostly by Philip Starr at UCSF. So this brings me to another part of my talk. So we talked about how we can have electricity, how we can put wires in the brain, and that we can do something. But why do we care? Why do we want to control the human brain? And there's 2 ways to think about it. The first way to think about it is, as I already said, we put it in, we turn it on, and patients get better. The second way to think about it is that arguably the only way to truly show our understanding of a system is our ability to control it. And a good analogy for that is ancient times, prehistoric times, where we had some early humans maybe observing lightning strikes, striking a tree or striking a piece of grass, starting a fire. So we can have fires, we can see what happens before fire, what after fire, and we can also maybe see the causality between the lightning strike and fire starting. But it was only when we were able to control the fire that we were able to get shelter, get warmth, cook food, get more calories from it, and that advanced the development of our brain and in turn development of our species. So control is interesting and helpful. So to formalize it slightly, we can talk about it in equations, but we can also talk about it in common language. So in common language, for example, let's say we are now in a room, and there is some temperature in this room. And we can say, OK, and we can just exist here. We can turn off AC, we can turn off heating, and we can just exist in this room. And over some time, the temperature may change. The sun may go down, it will get colder. And this is a system that exists autonomously. So this is the differential equation where we have some vector x evolving over time corresponding to some function f, which is a vector field. So when we start, for example, in one of those starting conditions marked with green circles, we can see that the system ends at a position driven by this vector field. And we can start at different positions and we can go back. And we can, for example, have stable limit cycles, like, for example, with seasonal changes in temperature. But when it gets interesting is when we can apply control. So now we have a control signal that can take us from one place in the vector field to another place in the state space using some input. And we can identify, using clever maths, the optimal trajectory. So what we can do, how we can adjust this input over time to get us from point A to point B. And then here we can see this expanded function where we have an input of the state that evolves in time. And then we have the control function that evolves with time. But you can see that this function is also only the function of time. It's not a function of the actual state that we are observing. And finally, we have the closed loop approach, which takes us from optimal trajectory, just one trajectory, to an optimal policy. So if we start at any point in this vector field, what is the policy that will take us from an arbitrary point to our destination? And here you can see the formalism changes, and our function is the function of a state. This can be a function of a state here, only takes the state in, and state is already dependent on time, or we can have a function that changes over time explicitly. And so for open loop temperature control, we can, for example, say we are going to turn heating from October to March, and we don't touch it. That's a good policy, perhaps, but then we may get too hot, too cold. So a closed loop approach would be, for example, if we had a thermometer measuring temperature in real time, and then turning on heating if temperature drops, and turning off heating or turning on AC if the temperature goes up. And that's objective closed loop. And a subjective closed loop would be, for example, asking everyone in the room if they are too hot or too cold, taking an average, and driving that closed loop system based on experience or based on the behavior or comfort level. And so we can take the same principles to controlling the brain. So if our understanding of concepts is driven by our ability to control them, then controlling the brain, which is the most difficult system in the world, is I would argue, the ultimate idea we want to do. So we have our brain state, which can be defined as, for example, functional imaging, like we had in the previous talk, or it can be defined by electrophysiology. We have some state evolving over time, and that state we can measure with some measurement function, and we can, for example, read out the brain activity. Also, this brain state can map to some manifold, and that manifold we can measure indirectly. And for example, here, the activity of the brain is measured through this manifold, through this other function, and we can, for example, operationalize it as seizure reduction. So we are measuring brain activity by how much the seizure burden changes for patients. So that was in open loop. If you want to, so that was autonomously. Now we can add this controller. So we implant the stimulator, and we can see those stimulation pulses delivered with some policy over time to treat the brain. And we can have the same readout again. Then we can close this loop. So now we have a closed system that takes some input from the brain, so for example, this brain activity measured some way, and adjusts the stimulation to what we observe in the brain. And that's the direct control. We can also have an indirect control when instead of measuring this brain activity, we measure some aspects of behavior. for example, seizure burden. And then we stimulate to reduce the seizure burden, not to reduce specific electrophysiological patterns in the brain. So NeuroPace responsive neurostimulation was the first such system that was approved in clinical care. This is what it looks like. We have the implantable pulse generator sitting flush with the skull. Then we have those wires, and we have two types of wires. We have strip electrodes, which sit on the surface of the brain, and then we have those depth leads that sit around the hippocampus and amygdala. And then we can drive stimulation in response to some patterns, then driving hopefully the seizure reduction. And we can see that this is doing quite well, so we get improved seizure reduction over time. But truly how we get to that seizure reduction is by adjusting the parameters of this device. But the problem is that currently it's not much better than putting the wire in, stimulating, and hoping for the best. We don't have a principled, scientifically grounded way of adjusting those parameters, which are currently by trial and error. So these are just some that we can change. We can change the pulse amplitude, width, or frequency. But that doesn't get us as far as we would like it to be. But this also brings us to the point that actually brain stimulation has always been open loop, closed loop. But this closed loop was not closed here in this space. It was closed by the physician adjusting the parameters on the readout, where we could see, for example, seizure reduction or some changes in behavior. For example, if we have stimulation for depression, improvement in depression scores. So where we actually took it, when the advancement was instead of having this connection here, we started having this connection from here. So having an objective measurement of brain activity and stimulating in response to that activity. And this is a classical case for reverse engineering. So we can stimulate the brain and we can see what happens, but that only drives us so far as understanding the specific stimulation and specific response that it causes. But reverse engineering, we observe behavior, we observe for whom stimulation works, for whom it doesn't, and then we can reverse engineer and we can walk back to our understanding of the system and we can take it apart and then we can reassemble it to get optimum results. And closed-loop simulation is inherently a reverse engineering tool. So for example, you can see it more clearly in this picture. So this picture is from deep brain stimulation, where we can see the electrode shaft with the contacts being inserted in a specific nucleus of the basal ganglia, the subthalamic nucleus. We have some sweet spot, perhaps, here where we want to target it, and we have some volume of tissue activated. So this is the tissue that's activated by the stimulation. In response to each stimulation, we can measure, directly or indirectly, some responses in the brain. And each of those stimulation pulses then becomes an experiment, and each of those responses becomes a new data point to improve our understanding of what's going on in this system, both mechanistically from a neuroscience point of view, but also for each patient, how we can have precision neuromodulation. And another dimension we can add is not just activating some piece of tissue, it's activating piece of tissue that's connected to other pieces of tissue around the brain. And that's truly what will take us to the next level. So we need to identify the right patient, the right place, and the right time for stimulation. And this is closing the third loop. This is closing the loop of development that takes years. So we had initially an idea that this responsive neurostimulation worked like cardiac pacemaker. So we detect arrhythmia, and then we stimulate, and then the arrhythmia goes away. It doesn't happen with seizures. We don't have evidence that this ever happens. What instead happens is we have long-term neuromodulatory neuroplasticity, and we have changes in the network organization and in the electrophysiological patterns we observe in response to the stimulation. So where we hope to be is identifying connectivity patterns that drive outcomes and our stimulation, location, so where we actually put those electrodes in the network, and state. So we want to be able to record brain state with a high enough temporal precision so we can maybe anticipate it, and instead of working retrospectively, stimulate prospectively in time. And this brings me to some research I'm focused on. So this is a busy slide, but it basically encapsulates everything I told you about. So we have some measurements of brain activity, and we can get some features out of it. We can have brain connectivity patterns, either functional or structural, from which we can have more features. And then also we can identify brain states, where perhaps some brain states are promoting good outcome and some of them may be impeding good outcome. And then we can combine it with modern machine learning techniques, and we can do cool stuff like we can phenotype patients. So we can identify subgroups of patients who may be more amenable to treatment or those who maybe are not amenable to treatment. We can predict trajectories of patient response early on in the treatment or even before the stimulation device is implanted, which is an amazing way to reduce neurosurgery effectively. We can predict who's going to be responder or non-responder. And we can also identify, we can reveal some mechanistic insights. So what is the most important feature in all of this that drives our outcome? Do we need to find the right patient? Do we need to stimulate in the right place? Or do we need to stimulate at the right time? So in summary, DBS or brain stimulation has always been closed loop. Just the loop was closed by the physician, adjusting the parameters in an iterative manner, not by objective measurement of brain activity. And this paradigm shift is mostly happening at the level of having faster feedback cycles instead of weeks, having millisecond or second precision. Quantitative optimization, so we can identify patterns, for example, using machine learning that promote good outcomes. And then finally, mechanistic discovery, so how we can do better in the future for future patients to optimize those therapies. That takes years currently. This is driven by the problem that trial and error tuning is not efficient enough. And this whole process will require combining connectomics, modeling, machine learning, and that's a collaboration between physicians, between neuroscientists, between engineers, and between companies who develop those devices. Right, thank you so much.